Good morning, everybody. Hope you're doing well. Happy Monday. Welcome to week 2 of school away from school. So this week, the work in biology class is going to be related to something called CRISPR, which looks like this. CRISPR. CRISPR stands for clustered regularly interspaced palindromic repeats, which is a really long word that means sequences of DNA that are the same forwards as they are backwards. And basically, this is a technology, and let's call it a technology, that humans have discovered in bacteria. And bacteria use this strategy to protect themselves against viral infection. It's kind of neat. And so what bacteria do is, you may remember from last week's video, if this is a bacteria and a virus lands on it and attaches to it, it inserts its DNA or RNA in there. Well, what the bacteria have evolved to do is make a copy of that viral DNA and insert it into their own DNA sequence, and then use that copied sequence to make a strand of RNA that then guides a protein in the bacteria to find the viral DNA and destroy it. And humans have adapted that technology basically to use to find and remove a sequence of DNA in any kind of cell. It doesn't have to be bacterial. And this is basically how it works. So take a look at this illustration, and I will leave this link on Google Classroom so you can explore this animation if you want to. Okay, so this is the CRISPR Cas9. It says mechanism and applications. Cas9 is this protein complex here, this sort of turquoise bit. Okay, and so what this is showing is this blue strip here is the guide RNA. So this is the piece, let's say this is how humans are using it. Humans will insert a piece of RNA into a cell, and they'll insert this protein complex, which is called Cas9, into a cell. And these pieces work together, and the protein, the Cas9 protein, carries this RNA transcript to the DNA until it finds the sequence of DNA that is complementary to it. What does complementary to it mean? It means if the DNA, if the RNA has an A, it looks for the T in DNA. If the RNA has a U, it looks for an A in the DNA. If the if the RNA has a C, it looks for a G in the DNA, etc. So let's say over in the animation, this blue piece of RNA has led the protein complex, the Cas9 complex, to the proper sequence of DNA. Well, then it opens that DNA and snips it right out of there. And there it cuts it out, okay? And then if a bacteria did this to a piece of viral DNA, then that viral DNA would no longer work. The way humans do it is we use that technology to snip out pieces of DNA that may be causing harmful traits, or we use it to do gene editing, meaning we snip out a section of DNA and replace it with a more desirable section. That's like to make a GMO. That's the kind of technology that's used there. Okay, so here is now it's just showing that comes back together. and has been altered somehow. Okay, so your job this week is going to be to learn about CRISPR in a couple of ways. There's a about 5-minute video, and then there's a sort of a diagram, and both of those assignments have questions with them, and that's part one. And then part two is going to be looking at some of the ethical implications of using CRISPR technology and getting your opinion about that. So that's how we're going to start the week, and I'll be hosting the chat time so we can have question and answer times. You can also always feel free to send me questions on email. But in any event, hope all is well with you, and hope you stay well.